Flora Grant Project, initiated and led by the Miawbogak First Nation, and today's presentation on honeybee evolution. Dr. Carr freely admits he knows nothing about honeybees, but can spot a faulty phylogenetic tree. He is the winner of 2 Editor's Choice Awards for work published in Genome. He once held an anti-war banner with Linus Pauling, and his retirement plans include death by twin teenage daughters in his 70s. And with that, you take it away. Oh, sorry, also a note that the session today is recorded, and so if you have any questions, they will also be recorded. That's all.
Okay, so I guess this, you can hear me, it's being amplified through there. Okay, what I want to talk about here is something that I've been working on for about 2 years now, and the background of this is kind of interesting. As was said in the introduction, I know absolutely nothing about honeybees except what I learned in introductory entomology 50 years ago. But this arose out of being asked as an associate editor to review a paper on honeybee systematics. And as I often do in these cases, I uploaded the data that they had supplied, started to compare it with some other data that were on GenBank, the repository for DNA sequence information, and found that there was a problem. And when I began to explore the problem, I found there was a bigger problem. And then when I got into it, I found that there was a huge problem. So I decided to reject the one paper and then go back and do a reevaluation of the data that were available and see if I could sort out what things happened. I've called the paper, or called the presentation and the paper, Things Fall Apart, because the previous theory about honeybee evolution is what's been called a out of Africa theory, essentially that honeybees evolved in Africa and then moved north. And what I'm going to do is suggest that that's 180 degrees wrong and that they actually evolved in Europe and moved south, and I'll present the evidence for that. Okay, we've had the land acknowledgement, but it is worth repeating. I respectfully acknowledge the territory in which we gather as the ancestral homeland of the Beothuk and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. I strive for respectful relationships with all peoples of this province as we search for collective healing and true reconciliation and honor this beautiful land together. The questions arise out of this one, and I don't know, I think this is still not working. This was sort of the theme. I was listening to a lot of Charlie Hayden at the time. And I'll play the music for you later. It's a great 3-voice a cappella song. Who you buzzing round now, Billy B, Billy B, who you buzzing round now? Why you buzzing around her, not me? And that really is the encapsulation of the question. Why are they in one place and not in another place? And how did they get from one place to another? I'm talking about exclusively here the genus Apis, which is, when you read the books and when you read the way it's used, Apis is the honeybees, 2 separate words because it's a kind of bee, and Apis is all of the honeybees, and nothing that is outside of Apis is a honeybee, and everything inside Apis is a honeybee. There are recognized morphologically 3 subgenera, Microapis, Megapis, and Apis. And these are, as you can tell from the names, the dwarf honeybees that extend over into Africa, Apis florea being the only one that extends to Africa, the giant honeybees, which are the ones in Southeast Asia or the global south, and then the cavity nesting honeybees, which extend up into less temperate climates. And Apis mellifera, which is the one that we're used to seeing in our gardens, is the only one that occurred originally in Europe and Asia, as well as into the Middle East. and it's the one that is exported worldwide as for making honey, for making wax, and also as a captive pollinator. And so it is the variation in Apis mellifera, the Linnaean species, that I want to talk about. Just to show some pretty pictures, here is the dwarf honeybee, and dwarf overstates it, I think, is that they're a bit smaller than the regular honeybee that we're used to, about 7 to 10 millimeters. And if you saw one, you would think, well, this is a small bee, but it's not all that small, not all that dwarfish. The giant honeybees are about wasp size. They get up to about twice the size of a regular bee. In a couple of these pictures, you can see the pollen sacs. And I think that if you saw one of these, or the dwarf honeybee, you probably wouldn't notice that it was any different than the bees that we've got. And I think certainly I wouldn't unless somebody directed this to me. And some of the pictures that I've got were sent by some bee biologists that I contacted in order that I wouldn't make any really overtly silly remarks. And here is our Western honeybee, also called the dark honeybee, also called the German honeybee. Goes by a lot of common names, and it's the usual thing where we don't use the common names, but we use the Linnaean name, which in this case is Apis mellifera mellifera. This animal is about 10 to 12 millimeters, and as I've said, this is the one that is used commercially. The only other species that is widely used commercially is Apis cerana, another one of the cavity nesting bees that is used for the same purpose in India, but that hasn't been exported beyond India. And a number of the local species are used locally in artisanal honey production. And to remind you how these things work, apiculture is the growth of bees in order for commercial purposes. We have the tagged queen in this case, so that the little tag is glued on. You can also put on a radio chip. This is the usual arrangement in a cave, as they're put in a mesh cage in order to keep them from escaping, because you want them to do work for you there. And then we have the drones, which are the males. And I'm not sure that I've ever actually seen a drone. I was curious to see that the eyes are very large and that the drones are stingless. And then we have the worker bees, which are female, and I'm not going to get into the whole haplodiploidy question here today. There are some questions that would come up later, but for now, we're just going to treat them as bees. And the relevant question to keep in mind is that every individual in a particular hive is an offspring of a single queen that's inside the hive. That varies by species. Honey gathering is very old. This is the oldest known example of it, about 8,000 years old, from the Aranya caves in Spain. And the drawing doesn't do it justice because there is actually a hole in the rock. And whether this is intended to be a honeybee cave, I couldn't find any information on. But you see that someone is climbing up the rock face or the tree. They've got a collecting basket and they are reaching into the hole or the cavity in order to collect the honey. We have documented pictures from the mid-15th century BCE from the New Kingdom of Egypt. This is a reproduction, actually. It was a drawing of a wall in the tomb of, I don't know how to pronounce this, Rekmire, and it shows that apiculture was very well developed. You see that the upper worker has exactly what is used today, is that there is a smoke bowl that, and these little things direct smoke into the artificial hives that pacifies the bees, and then the other person down here is collecting not pieces of bread out of the oven, but is collecting combs, and the regularity of the combs suggests to the people that are commenting on this that these are probably artificial combs. So by 3,500 years ago, this is a very well-established art. Bees also get into the news as the Africanized bees, or the so-called killer bees. The African bees result from a cross between two of the subspecies that I'll talk about, Apis mellifera scutellata, which is a South African bee, and Apis mellifera ligustica, which is the Italian honeybee. It's a variant on the more northerly Mellifera mellifera. The story on this one seems to be, because there's been a lot of cover up, as far as I can tell, that either a breeder in in Brazil, knew that the scutellata gave more honey, and so imported some queens without a permit, and that those queens escaped and crossbred with the local hives, and that produced a bee which did in fact produce more honey, but it also produced a much more aggressive bee. And the beekeepers are perfectly happy with that because they're wearing gear. But the swarming activity of these and the aggressiveness of the bees does make them killers, especially if you're a small child or a small animal. And I won't say anything more about that. The other alternative theory is that they were actually imported by a laboratory that was intending to do this as a scientific experiment, and the laboratory was not secure, and the animals escaped from a laboratory. So there's, who do you want to blame? Which side are you on? What have people thought about the relationships between subspecies of mellifera? Now, unlike most of the other honeybee species, there's at least 24 recognized and named species of Apis mellifera in the literature. And this goes back to the 1700s. People have added them on. European biologists are especially good at this. There's multiple subspecies have been named in Europe, Asia Minor, the Levant, and once Africa was invaded, more species were found there. The consensus as of 1988, and using the old methods of doing systematics, where Rutner mostly did measurements on wing venation. And having done a little bit of work with insects, I know that the capacity to try to measure discrete things about wings and about body shapes influenced the way that people set up classifications. This is a phenogram, and by a phenogram, it's simply attempting to separate different sorts of bees. And Rutner by no means was attempting a modern systematic relationship. He was simply dividing them into groups. And what he made out on these is what's been called the ACMO system, the ACMO system. And this says that there are 4 subspecies groups on separate continents. So you'll notice that this diagram is like a jack. Each of the 4 legs is directed to a different continent. A is for African, C is for continental, which means European. M is for mellifera, taking the type subspecies as recognizable. And O is for oriental. A better name for that one would be Asian, because these bees do not go that far east. They go into the Levantine region, and they get as far east as Saudi Arabia and maybe into the Caucasus. So this was the situation that was faced by the people that first started using DNA to look at these things. So we got 4 groups, and we want to look at it by modern methods, and the modern methods are phylogenetic systematics. And for those of you that have not taken my classes, this is Phylogenetic Systematics 101 on the origin of subspecies. And whenever you have 4 groups, so A, B, C, and D, 4 taxa, and those could be subspecies, they could be species, they could be species groups, they could be genera. You have the 4 taxon problem. And the 4 taxon problem is solved by a 2-step process. The first thing that you do is use your data, and it could be DNA data, and it could be other kinds of data, but think of this in terms of the DNA data, is first you want to determine what is one of the species' closest relatives. So you figure what are the pairwise relationships. So relative to A, B could be the closest relative, meaning that they have a common ancestor, or C could be the closest, or D could be the closest. So you'd want to make that determination. Having made that determination, and supposing for present purposes that you've decided that A and B share a recent common ancestor than do C and D, and so C and D also share a more common ancestor, then you have this diagram, which we will call a network. Okay, and this is a network. We can't call it a tree, because what does a tree have that a network doesn't have is a tree has a root. So you have to know what the origin is. So then you need to place the root of the tree, and you ordinarily do this by using an outgroup, and ideally an outgroup would be a taxon that is outside, is known to be outside on good authority, is known to be outside the group of animals that you are working with or plants. And ideally should be the closest relative, not included in the group. But since you often don't know that, anything outside the group within reason will work. Okay, then the 5 possible rootings are that you could place it on the A branch, the B branch, the C branch, or the D branch, or you could place it on the internode between the 2 pairs. And this would give you 5 possible phylogenetic trees. And if you look at these, they all have different implications for how these critters are related. And here, A is the outgroup to B, C, and D, which is to say that B, C, and D have a more recent common ancestor than any does to A. C and D have a more recent common ancestor than either is to B, the 4 taxon statement. And then we could also make that with respect to B, with respect to C, with respect to D, or if we root on the internode, then A and B are a pair and C and D are a pair. So the method for doing this is quite straightforward. You can read about it in any of the books, works very well with DNA data. Okay, and the problem that we are trying to resolve, given that people that have come along afterwards have sort of agreed, and I'm going to speak to this as being what I think is one of the issues, the continental arrangement of African, European mellifera and Asian or Oriental seems to influence people that are working on the group very heavily. Ruttner is very highly regarded. Informally, you can ask me afterwards some questions about Ruttner. And the Whitfield et al. In 2006 came up with the first nuclear DNA network. And they presented their data in a way that wasn't really phylogenetic, in my opinion. You can look at the paper and you can see their groups, but there is not a specifically phylogenetic arrangement. Hahn in 2012 took exactly the same data and did treat it in a phylogenetic manner. And the essence of their phylogenetic arrangement was, well, we're going to put in an outgroup. And as the outgroup, we're going to take Apis cerana, which, as I said before, is one of the other cavity nesting bees. And according to this rooting, using the same terminology I used before, the root is placed on the internode. And the suggestion is that the African forms go along with mellifera, the continental and Asian forms go together. And they think, Han thought that, well, this favored an Oriental origin, and you can sort of shove the internet over here. Or if you want to go with Whitfield sort of put the internet, or put the note that, excuse me, the root here. And then one of the questions is exactly where you put it in here, where these things are genetically very similar, you're going to get a different result. Okay, so based on Whitfield's data, the tree would be rooted so that the African group is the original, and then things came out of Africa, went to Europe. They went to, actually went to Europe twice. They went to Asia, and then there's a third invasion of Brazil, but we won't worry about that one. According to Han, they want to place the root in the Oriental group, and then you would get into Africa, and after that you would get to Europe. So any of these things place Europe as the last place that is invaded by these bees, and most of them say that it's got to be in the global south. I don't expect you to pay attention to the subspecies names, so I'm just going to make this biogeographically a little bit easier. When we look at the 13 subspecies that were examined in this work, they're from South Africa, they're from North Africa, they're from Sub-Saharan, these are Sub-Saharan animals. They're also from the Nile Valley, Nilotic, they're from Ethiopia. Mellifera is basically mellifera and one other subspecies that's found in the Iberian Peninsula in Spain. The Eastern Europe species are Italian and a group of very narrowly distributed Eastern European bees. And then the Asian bees are from the Caucasus, Kazakhstan, Asia Minor, Turkey, and the Levant, which is Syria. And you're going to see one of the problems here is that putting the Caucasus, Kazakhstan, Turkey, and Syria, and calling that all a coherent Asian or Oriental group, that's going to cause some problems because those animals really aren't close to each other. So we'll return to this issue. Okay, so what I did is a meta-analysis of existing data, and the advantage of a meta-analysis is that you don't get your hands dirty, you don't have to have a grant, you're inside in winter and there's no heavy lifting. You just have to have a computer that's high speed. This is reported in Nature Scientific Reports back in June, and I simply went to GenBank, which is the repository for all of the DNA sequences, and I downloaded all of the complete mitochondrial DNA genomes associated with the genus Apis, and I did that through October of 2022. There's some other things that I could talk about that are extensions of that, but this is basically what the paper is based on. The data that I used were only the coding regions. I've eliminated the ribosomal DNA regions, the transfer DNAs, and the D loop. And the basic reason for that is that those three regions are very difficult to align across even closely related subspecies. Certainly when I tried to do it between species, in many cases, I just threw up my hands because this can't be aligned, and any alignment that you would do is completely arbitrary. If you stick to coding regions, you have the triplet framework as a backbone, and the lengths of the genes, with some minor exceptions, are basically the same across all of the taxa that we're dealing with. So this just made it much easier to have a data set that was coherent. It includes 13 genes, 9 on the heavy strand, 4 on the light strand, for a total of just over 11,000 base pairs out of the 16,000 base pairs that are in the molecule as a whole. The taxa that I used, first I chose an outgroup. Bombus ignitus is one of the bumblebees. The bumblebees are not necessarily the closest group to the honeybees, but there are a lot of bumblebees. I tried this with several bumblebees and I always got the same results. I used 9 species of Apis outside Apis mellifera, for which there are complete DNA sequences. I'm extending that work. There's probably about a dozen species outside of mellifera. And then for Apis mellifera, I used a total of 78 individuals representing 22 subspecies in the literature. And an important part of this work is that I had multiple replicate sequences from 8 of the subspecies. And one of the problems that was apparent to me very early on in doing this is that what one person would call Apis mellifera scutellata was not what another person would call that, and that something like Intermesa, I think was the one that I first saw, they were just wildly different, so they couldn't be the same subspecies. So I wanted to get multiple representatives to see what was going on. This goes to the question of alpha taxonomy. Alpha taxonomy is the thing that is really basic, is getting the names right. If you collect an organism, you should be able to assign it to a proper name. It's called keying out if you're doing it in an introductory class. And this assumes that the basic taxonomy of the group has been sorted out. The other problem that we run into with alpha taxonomy is GenBank curation. The people that are curating DNA sequences in GenBank are, for the most part, molecular biologists who do not appreciate the importance of proper taxonomy and proper curation. And GenBank, unless you correct them and unless you almost yell at them, they will accept whatever name a person assigns to them. So again, when you're dealing with molecular biologists who catch a bee, and they're faced with the choice of, well, what am I going to call this bee? Let me look in my college book, and it says that Apis mellifera is Apis mellifera mellifera, so I'll put that name in with it. So without necessarily knowing what they're talking about. In the old days, you had problems. If you wanted to know the information that was available on the domestic cat, the problem was you had the correct name of the cat was Felis catus, C-A-T-U-S. Some people would put it in under Felis catus, C-A-T-T-U-S. You wouldn't catch it if you didn't look under the right name. And the very worst one was some people would put it in under Felis domesticus. And the only place that a Felis domesticus is the name of a domestic cat is in a Bugs Bunny carcass. So it was a real mess. I was on a committee that tried to get them to sort that out. Phylogenetic methods are the ones I won't go into detail. I use 3 different methods that take 3 different approaches. Maximum parsimony, which is seeking to minimize the number of SNP changes, the number of mutational changes that occur in a tree. Neighbor joining, which is a phonetic method, and you simply measure the difference between 2 sequences by saying what is the number, how many base pairs did they differ by. And then maximum likelihood is a Bayesian method, and this is based on having a model for how evolution would occur in your groups. You can estimate this empirically from the distribution of the bases. And the nice thing about having a large data set is that the 3 different methods should converge on the same answer. And in fact, they do. So I won't talk anymore about the 3 different methods, and everything that you're going to see is based on a maximum parsimony, because that was a way of getting at some of the other questions. Statistical confidence is a bootstrap method. You resample the data 3,000 times. So given that on the assumption that your DNA sequence for an individual is correct, you say, well, if I were to redraw with replacement 11,006 base pairs, would I get the same tree the next time I do it? And I asked that question 3,000 times. So it gives you confidence on the trees. And the last thing was to put in a molecular clock to attempt to use the data to estimate times. And the standard way of doing this is with a linearized maximum likelihood, because the model-based is probably the most accurate for this. You linearize it, so you force all of the branches to come out at the same end. And then I used here, and this is the least satisfactory part of the whole paper, the standard insect rate, 0.0115 substitutions per site per million years. And to do this properly, there would have to be a clock that was calibrated for bees, and there simply isn't the information available to do that. I'm looking at the bee form of DNA. Is anybody going to laugh at all if I talk about this as bee DNA? No, okay, so there's nobody from my class. This is what the data looks like, and I put this in case you're easily impressed, just short of 1 million base pairs, and I include this, and there's also, this is also what the DNA looked like, so if you want to come and look at a 3D model of DNA, that's there. 957,522 base pairs, 1 million base pairs. Took me a couple of weeks to put together. My PhD thesis was based on 106 DNA markers and took 3 years. So things do evolve, things get better. So you just collect all the DNA, you drop it into a program, and the program will allow you to do the analyses. Okay, so I do the maximum parsimony, and as I've said, you get exactly the same results if you do it by another method. And what you're looking at in this tree is the numbers above the branches are the number of SNP differences. We won't call them mutations because they didn't occur. They're not mutations. There are things that have happened in the past, and now they're simply showing up as differences between the different organisms. So the number of changes that occur on each branch, notice that a typical pair of species is going to be separated by 300 plus 300 by about 600 substitutions out of 11,000. And the answers that we get here, and this is nothing new, it simply confirms what was already known and what other people had done, is the dwarf honeybees are at the base of the tree. Next come the giant honeybees. So these are our pairs. And most recently come the cavity nesting honeybees. The arrangement of these agrees with what was done before, except that this is a tree-based ascertainment that Apis mellifera mellifera is at the base of the tree, whereas the prejudice would ordinarily be to say that the European one is the most advanced. So a lot of the early work that's done says, well, mellifera is at the end, and then we compare everything to that. And here, Apis mellifera is at the bottom, and then we compare everything against that. And then again, the pictures there, Bombus ignitus on the outside. Okay, so what this establishes is the position of mellifera within the tree, and it also establishes that Serrana is an okay outgroup to use with mellifera because it's part of the same cavity nesting honeybees. And the results that I'm going to show you, I checked them against this species, against this species, against this species, and against all the species together and in various combinations. So the results do not depend on the particular outgroup. This is the simplified version for people who are interested in evolution but don't want to worry about the names. And what I got is this part of the tree shows you the first half of the results, and this is 18 Eurasian and Afro-Asian species using Eurasian and Afro-Asian in a very broad sense, trying to keep the names that Rutner and Whiteley used, but looking at them in a new way. And when you do this, much to everyone's surprise, much to my surprise, checked it repeatedly, Apis mellifera comes out at the base. So the European species comes out at the base of the tree. Apis mellifera is quite diverse. And an interesting thing is that this is from Western China. And the difference between this Chinese bee, which is morphologically quite different, the difference between this one and a type mellifera is much less than the difference between different complements of, or different specimens of mellifera. So the North European form comes out first. Then when you look at the rest of them, they come out into 6 very well-defined clades. And one of the clades I will call Eurasian, and it splits nicely into first a Southeast European group. The Ligustica, as I've said, is the Italian honeybee. There's quite a number of different representatives. And this would be an example that I won't talk too much about, but if you look at the variation amongst what is called Ligustica, the variation within this subspecies is greater than the variation between 2 nominal subspecies that are represented by a single individual once. So in the language of systematics, I would suggest that Apis has been oversplit. These are recognized morphologically. Whether they are really genetically distinct is another question. The Asia Minor species, again, the same thing, less variation between these 3 species from Persia, Medes, Anatolia, Turkey, and Caucasia, the Caucasus, less variation amongst these 3 species than we find within Ligustica. But the Southeast European and the Asia Minor species are very closely associated. We then get into a Levant Near Eastern group, and the Levant is very broadly defined as Eastern Mediterranean, and it would go down to into Egypt, it would go up into Asia Minor, it would go as far east as Iraq, depending on who you read. So it is a convenient thing to say it's Levantine and Near Eastern. And here again, there's a huge amount of variation in the most common species in that region, Yemenitica. And the reason that it's called Yemenitica is that it was originally described from Yemen on the Arabian Peninsula. But it's also found up into the Levant. It's found in Asia. It's found very closely related to the Nile Valley form, is Lamarcki. And then here's a true Levantine form, one found from Syria. Okay, so there's a whole swarm found in the Levant. Then we start to get into the African species, and these are Simensis, which means coming from Ethiopia. Unicolor, because they're all basically a black color. Unicolor is, the type locality is the island of Malagasy. And so you'll see that we are progressing southward, if you think of the Nile Valley, and then Ethiopia, and then Malagasy. And then only at that point do we get into sub-Saharan Africa. But we've also got a sub-branch, and it's a sister branch to this one. It doesn't derive from the South African one, that I will call the Mediterranean. And these are found around the Mediterranean and also in the Mediterranean on some of the islands. And again, this shows you the difficulty of being really typological about what you call a continent. I'm going to show you, or I am showing you, that this group includes something that's found in Iberia on the north slope of the Mediterranean. It includes things that are found in oases in Africa. They're very closely related. Things that are found on Crete, things that are found on Malta, Ratnarai. And so whether you call those islands European or African, and whether you say something that occurs on the margin of Mediterranean, African, or European, you're not paying attention to the big picture. You're trying to stick them into one continent or the other. Okay, this leaves us with the next part of the tree, and there's a lot of information available on Scytolata, which is the standard sub-Saharan African bee. and capensis, which was originally described just from the Cape. And it turns out when people go looking for it, that forms that are like that are found further north. The dividing line between capensis and scutellata is not at all clear. One of the major problems that you can see here is if you look at the things that I've labeled capensis and scutellata, yes, you can make out a capensis clade here, and there's a scutellata. Somebody's identified this as a scutellata, fits into that clade. And even worse, within scutellata, there's a number of capensis, and even worse, within scutellata, there are two discernible clades of scutellata, and I won't try to prove it here, but they occur in different places. But the problem with them occurring in different places is that somebody that is looking for bees, they go to one place and all the bees in that area look alike, and then somebody else goes to another place and all of the bees that are there look alike each other morphologically and genetically. So you don't know, are these two separate species and just nobody has bothered to separate them, or are they really part of the same species? And if I looked other places, would I see more complicated pictures? We also have the West African bee, Adansonii, and the same issue. Only one of them has been looked at, even though this is a very common form in Western Africa and Equatorial Africa. And we also have Monticola, which is found in the mountains of Kenya. And here's an animal called capensis. And I suggest that it's easier to think that this is a misidentified Monticola. When you go back to the paper, you do find that problem. So this gives us a phylogenetic evolution of honeybees. The other literary title that I could use here is The Scramble for Africa, which is a very nice book written on what happened in the late 19th century. Is all the European powers decided, well, Africa is the last great unexploited frontier. So we're going to sit in the conference room and we're going to divvy out all of the land in Africa without paying any attention to the fact that it's already owned by other people. So we'll just assign it. So this is the scramble for Africa, and that European bees are scrambling for the South. The pattern then is that we have, according to my theory, that we have Apis mellifera. Mellifera is well established in Northern Europe. Upper limit is Norway and southern Sweden. There is a expansion into southeastern Europe, and each one of these dots is a separate subspecies. And you get across the isthmus there, and then you're into Asia Minor, so Anatolia, the Caucasus, and into Persia, the old name. Then you get into the Levant and the Near East. They are further east than that, but here's our Syrian form, here's our Nilotic form, here are our Yamani forms. They're also found down here in Kenya, where somebody did a very extensive survey. Then the next thing south is Ethiopia. And the next thing south is the island of Malagasy. The books tell you that unicolor is confined to the island, and that's just because you haven't looked hard enough. And again, when my friend here looked at a bunch of bees from Kenya, there are the unicolor form there, as well as the Levantine form, as well as Monticola, as well as what is identified as scutellata. And something that I want to talk about is they're completely separate from everything else that's called scutellata. So we've probably got a new subspecies, and one begins to think that bee biology is not as mature as perhaps we would like to believe. Scutellata and capensis mixed together at the Cape and northwards, and then the Mediterranean. And when you look at the tree, the Iberian population is first established. Then you have oasis forms in Tunisia and Morocco. And then you have things that are found on the west end of Sicily and also on Crete and Malta. And when you look at some of the other data, you find that the original European form is found on the east end of Sicily. They meet in the middle of Sicily and they don't appear to cross. So you have two different kinds of bees from different lineages meeting in the middle. And also when you look at the other islands in the east, these are also the European forms. So the reinvasion is from the west and the other ones occur in the east. So biogeographically, it's a terrific hypothesis because everything just goes in a straight line. If I put a time scale on this and not having an enormous confidence in the time, as I've said, that we get successive establishment and that you can look along the backbone of the tree, I'm reasonably confident that the origin of mellifera is around 800,000 years ago. Some of the fossil people don't like that date. And then there is a series of establishments as the animals move south over the next 500,000 years or so. And then there is diversification within each of those regions that are established. And they're on the order of 140 to 200,000 years ago. And we have local groups for only a few tens of thousands of years. Okay, so the discussion of all of this is, I've told you my side of the story, what do other people say? And with Winnie the Pooh, I say that I have just been thinking and I have come to a very important decision. These are the wrong sort of bees, by which I mean your bees are the wrong sort of bees. The one that I spent the most time on, and this is where the taxonomy problems were most obvious, were two studies made by Boardman and Emanuel at Florida. And this is not to criticize them because they were the ones that generated this data over the space of 20 years. They would crank out a new sequence and then they would crank out another sequence. So the fact that we have these data is due to these folks. Eric Tehelka did what I'm doing here. He took those same data and reanalyzed them. And if Boardman and Emanuel came up with a wrong conclusion, problematic conclusion, then Tehelka did even more so. The problems I've shown are the ones with Asteraces and then also this problem here. So depending on whose intermesa you look at, and this is intermesa is one of those oasis bees from the north, it's either up here with the Levantine animals or it's down here with the other animals that occur there. Okay, so one of these sequences is clearly not right. And if you look at the standard things that you would look at, you see that Syriaca and intermesa are guilty of long branch attraction. And what this suggests is that the sequences have gotten mixed up. So this isn't this really isn't where they are, but intermesa pulls Syriaca out and Syriaca pulls intermesa out. And then when you look at the next one, the accession numbers are in order. So these two species were done at the same time. Okay, so the correct species to use would be Syriaca down here with the Levantine species and intermesa up here with the Mediterranean species. The other problem that's true in both cases is that when you choose Scutellata and Capensis to show the African forms, Boardman got this right. And but they used single specimens. And as it happens, their Capensis is from the broader Capensis clade that I found. And their Scutellata is from the broader, but only one of the Scutellata clades that I found. And then if we look at Tehelka, Tehelka had the extraordinary bad luck to choose, yes, a proper Capensis from the same clade that these folks did, but their Scutellata is one that is labeled scutellata, but probably isn't, and it's actually a capensis. So what they get is capensis and scutellata are very closely related. And then the two that we're really interested in, which are Adansoni and Monticola, they occur outside that group, when actually the larger data show that Monticola are part of the capensis group and that Adansoni is part of the scutellata group. If we go back to the nuclear comparison, the only thing I will do here is show you what the problems are. So again, this is that same diagram I showed you at the beginning, organized by geography. One thing that you see is that my data in the green boxes put the Italian and Eastern European species together with the Caucasian, Kazaki, and Anatolia. Those regions are contiguous with each other, and the nuclear DNA put them in completely different groups. And again, I think partly because you think that there's a continental group and you think there's an Asian group, your interpretation is going to be biased in that direction. Similarly, well, the Nile Valley is clearly African, the Levant is clearly Asian, but if you look at the Levant and the Nile Valley, they're only a few miles apart, and the Levant is geographically very close to the Nile Valley. So you have to look at the contiguity and the path between those, rather than looking at the fact that one is nominally Asian and one is nominally African. And then the other one that is really apparent is there are the North African oasis bees on the southern coast of the Mediterranean. There is the Spanish bee on the southern coast of the north side of the Mediterranean. And these are only a few hundred miles from each other, but that problem of saying North Africa is part of Africa and Iberia is part of Europe, my data say that those are essentially identical. The interpretation here is that they are on separate continents. I see that I'm running out of time, so this is just, if anybody wants to look at this, and I can give you the paper, this is just reviewing what I said before. The sources of error are what I wanted to emphasize here, and the sources of error in the papers that are using single specimens to represent a species. The problem is that if you get that wrong, everything else is garbage after that. So you have to choose your, you have to get multiple replicates to make sure that you've got the actual species. The paraphyly artifacts, so saying that two things that appear to be very different from each other are the same species, those are artifacts of misreferral in GenBank. And the most glaring case of this is that my friend who worked in Kenya gave the sequences to his graduate student to submit to GenBank. And the story that I'm getting is the graduate student didn't quite know how to do that properly. And when the graduate student picked the name that they were to go under, he chose from the drop-down list. And the first one there was Apis mellifera mellifera. So they all went in under that, even though it was known that they belonged to a different set of species. And so I had to call him up and say, hey, did you know that there's a problem? And he said, yeah, but we got it right. And I said, but it's not that way in GenBank. And he said a bad word and has promised to fix it. You get problematic inferences if you're using faulty sequences. So you actually need to check your technical data. And my note is that DNA does not lie, but GenBank is not infallible. Curation is sometimes flawed. And depending on the maturity of the taxonomy in your particular group, you'll get a different result. Melittologist is a word that I learned only yesterday. A melittologist is somebody who studies honeybees as opposed to an apiculturalist. And the lesson for this one is a good expression from Texas. I don't know anything about this, but I'll tell you what I think. I don't know anything about honeybees, but I do know how to spot errors in a phylogenetic analysis. Always get a second opinion. Don't assume that the animal or the sequence you've been handled is correctly labeled. Don't believe everything you read. So when you go to the papers and somebody says, well, we're calling it here and it came from here, so isn't that interesting that the range is extended over there? No, it's not, because it came from over there. It's something that belongs there and you just got the name wrong. Names matter. As I've said before, alpha taxonomy is still vitally important. If you don't get your names right in the first place, you're going to have problems. And DNA does not lie, but duration is sometimes flawed. And as Alice said so long ago, I don't think they play it all fairly. Alice began in a rather complaining tone. And they all quarrel so dreadfully. One can't hear oneself speak, and they don't seem to have any rules in particular. At least if there are, nobody attends to them, and you have no idea how confusing it is, all the things being alive. So if we were physicists, we wouldn't have this problem, but we're biologists, we have this problem. So thank you.
Thank you, Dr. Carr. So we're a little bit past 1 now, but if there are any burning questions, we will accept them.
Steve, have you thought about, or are you moving towards some biogeographic work? I was imagining that, you know, with a, what is it, 900,000 year story, that perhaps glaciation plays a part in this, or?
I think that glaciation may explain European distribution. To say that those things, those subspecies are very, very close. So the human were like the other out of humans getting into the Near East 7,000. Well, humans are going this way, and the humans it's what are the windows get into last 1,000 years, which is my answer to a lot of things. I think the phylogeography is critical here, and folks would get away from the idea that continents per se are running things.
All right, well, thanks again, Dr. Carr. And I think there's still some coffee and cookies left. Oh, did you have a question? Okay. All right, thank you.